All right, hello class. This is the second lecture on the blood, okay? Blood is the first of the three major parts of the cardiovascular system, the blood, the heart, and the vessels. And so last time we dove into the blood. What is blood composed of? When we take whole blood and we spin it down in a centrifuge, we can begin to separate or fractionate the blood into its component parts, okay? 55% of which is plasma, which is kind of fancy water with a bunch of proteins. We looked at several proteins before, and then these different smaller molecules or ions, glucose, amino acids, fatty acids here, okay? So we looked at plasma and all of its components, and we started to look at the formed elements, okay? Formed elements are the cellular derived parts of the blood, okay? The red blood cells, the white blood cells, and the platelets, okay? We started looking at red blood cells in detail last time. We looked at the shape of the cells, the size, hemoglobin, anemia, and we began to look at cellular turnover and kind of the life cycle of a red blood cell, okay? We're going to continue talking about that, okay? Again, three kind of analogies for these formed elements. Red blood cells are kind of like a taxi cab filled with sponges, okay? The white blood cells are kind of like a sentry or a guard or a policeman watching out for bad guys that might want to invade our body, okay? And then we have platelets, which function kind of like a plug to stop blood flow when there is a break in the tissue. Okay, went through some general characteristics, the proteins, okay? Talked about the properties of red blood cells, we talked about hemoglobin, talked about the different ways in which anemia can affect the capacity to carry oxygen. And we talked about fetal hemoglobin has these two different genes. The gamma subunit is present in the fetus, and during development it will transition to the beta subunit. All right, so this is really the topic that we're started to address, talking about where do blood cells begin, where are they born, and then they live their life floating around or circulating around the body for about 120 days, and after that they go to the spleen, okay? And then they turn over. The parts of those red blood cells get recycled, some of them get excreted. So in this diagram we talked about the protein of the hemoglobin, because red blood cells are basically a sack of hemoglobin, as I like to describe it, okay? It's just a one cell that has really not a lot of organelles in it, okay? They don't have mitochondria, they don't have other organelles, they just have a bunch of hemoglobin. So that protein, the hemoglobin, must be recycled, okay? That has a certain pathway. We talked about the iron. Iron is a very unique metallic element in the body. We don't have a lot of use of, iron is used a lot of other places in the body, but it needs to be bound to something. So when we talk about how iron is carried around the body, we're going to be talking about another carrier protein called transferrin, which transports iron from the spleen to the bone. And then we talked about heme, okay, and how heme, again, heme breakdown starts in the spleen and it continues in the liver, and then the liver processes it into a form that we can excrete either in our stool, number 2, or in our urine. If we have liver problems, then we can have jaundice, which is this accumulation of this yellow pigment called bilirubin. All right, so let's continue talking about red blood cell production. Where do red blood cells come from? Okay, where are they derived from? They're derived from a special type of stem cell that is in bone marrow. Okay, we talked about bones last semester in A&P 1. Bone tissue has compact bone and spongy bone. And in the spongy bone, in between all of the trabeculae, or these cross bridges of trabecular bone, we have red marrow. Okay, red marrow contains these stem cells and a bunch of developing cells for white blood cells and red blood cells and platelets. Okay, so this whole process is called erythropoiesis. Okay, erythro is red, poiesis means the making of these cell lineages. Okay, so here's a big mega diagram. Remember, we're focusing here on red blood cells yet, and so from the precursor stem cell down to the bottom, we have this process of differentiation. Okay, differentiation is a sequential change in a cell's makeup that specifies it for function. Okay, let me say that again. Okay, differentiation is a sequence of cellular events that specifies or the cell gets specialized for a particular function. Red blood cells have a very different function than platelets that have a very different function from leukocytes. Okay, so differentiation is that process. Okay, along the way there are different branching points that are controlled by other hormones. Okay, a hormone can signal a differentiation step. Okay, so we're here talking about this particular pathway. How do we end up down here with a mature red blood cell? Well, we start off with what's called a hemocytoblast. Okay, it's called a hematopoietic stem cell. Hematopoiesis is the differentiation of all blood cells, white blood cells and red blood cells and platelets. They all derive from a common stem cell. Okay, all of these blood cell types derive from a common stem cell. And that stem cell can then differentiate based on what it's exposed to, okay, along the way. Okay, so here we have the pathway for making red blood cells. Along the way, I want you to notice here, if I back this up so you can see this a little bit more clearly, is there's an important hormone here we're going to talk about called EPO. Erythropoietin is the name of that. EPO is the abbreviation. EPO stimulates the myeloid stem cell line towards this lineage, this pathway for maturing the red blood cells. Okay? So early on, it has a nucleus. You can notice that here. It looks kind of like just a vague blob of a cell. Okay? The blue cytoplasm and the purple nucleus. And along the way, it becomes more and more specialized. This cell begins to produce lots and lots of which protein? Hemoglobin. Okay? It's going to make lots and lots of hemoglobin. In the process of that, it turns red because hemoglobin is red, has a red pigment, which is hemoglobin. Along the way, that nucleus starts to get smaller and smaller because the role of the DNA making the instructions for that protein, it's all done. Okay? This cell is now completely full of hemoglobin, and there's really no function for that nucleus any longer, so it can literally spit it out of the cell, okay, eject it, and that will become reabsorbed by other white blood cells, okay, become phagocytized. And then the resulting cell, which is called a reticulocyte, okay, it's kind of a precursor red blood cell. The reticulocyte will become the red blood cell. Alrighty, so here's that process, okay? Day 1, day 2, day 3, day 4, 5 through 6, okay? So about 7 days to differentiate from the stem cell down to a mature red blood cell. Okay, what do we need to make a red blood cell? Okay, we need amino acids because we're making a lot of hemoglobin. Okay, we need iron because we need to insert an iron molecule into the heme group of the hemoglobin. And we need a very special cofactor called vitamin B12. Okay, vitamin B12 and B6 and folic acid, also important. But especially the B vitamins are very important for rapidly dividing cells. Okay, DNA synthesis requires these cofactors. Okay, rapidly dividing cells require these cofactors. So there's a form of anemia called pernicious anemia. Pernicious anemia is linked to a B vitamin deficiency, typically vitamin B12. If you don't have enough vitamin B12, then it can lead to a slower response time in making new red blood cells. So people can have anemia because they just have low vitamin B12. Okay, so they might have to supplement that in unique ways. Okay, so rapidly dividing cells, which red blood cells are being made constantly in the bone marrow. Okay, and it's sensitive to even small decreases or deficiencies in the B vitamins and folic acid. So let's talk about EPO. Okay, erythropoietin. Erythropoietin, we said, was an early hormone. Okay, here, okay, it's a hormone that's stimulating this precursor cell to actually continue to differentiate into a mature red blood cell. Okay, so erythropoietin, that's the hormone. It's secreted under conditions of low oxygen. Okay, low oxygen, the name for that is called hypoxia. Remember hyper and hypo? Hypo means low something, right? Hyper is too much. So hypoxia, okay? There's no additional O there. Oxia is oxygen, so low oxygen. So it's detected, low oxygen is detected in the kidneys. Of all things, you think, well, why doesn't it detect that like in the lungs or somewhere else? But the kidney turns out to be a very good place to monitor oxygen levels. Okay, here's the bloodstream bringing lots of blood into the kidney, because the kidneys at any one given time are processing about a quarter of all the blood in the body. Okay, all the time the blood is circulating, and about 1/4 of the volume of our blood is in our kidneys. Okay, so it's a constant sensor for oxygen. Okay, so blood is flowing into the kidneys all the time, and it's removing waste and producing urine. Okay, so this tube here would lead down into the urinary bladder to make pee, okay, make urine. Okay, dirty blood in, clean blood out. Okay, so in the process, it's sensing for oxygen levels. Okay, and so if it's deficient in oxygen, well then the kidney is a secondary endocrine organ, has a gland that can secrete erythropoietin. What does erythropoietin do? We just looked at it. It stimulates this pathway, okay, of differentiating the stem cells into the mature red blood cells. So this here on the side is just the same pathway as what we've seen before. Erythropoietin is signaling and allowing for that pathway to move forward, okay, and actually stimulating the growth and the differentiation. That growth will then stimulate more red blood cell production. Okay, so if we have low oxygen being sensed in the kidneys, okay, EPO will increase red blood cell production. If we have plenty of oxygen, the kidneys will detect that and will slow down erythropoietin and will slow down the production. It's not like off or on, it is more like a lot versus a little. Okay, there's always a little bit of erythropoietin being secreted all the time. All right, so question, why do red blood cells have dimples? Okay, well, we talked about before that they makes them very flexible, gives them a high surface area, and allows for rapid gas exchange. Okay, so that's the functional reason for that. But the structural reason is because of a protein called spectrin. Okay, this is just kind of a trivia thing. This protein spectrin is arranged in a very particular order, and it allows for this dimpling effect to occur. Okay, spectrin is a protein that kind of pulls the dimple inward in this circular form. Okay? You don't have to memorize where it's located, but spectrin is the name of this protein that gives it this dimpled appearance. Kind of interesting. All right, let's talk about blood typing, because as we might be aware, that blood typing is important, because if we have a need for blood, because we're always, having accidents and going to war and stuff like that. Okay, so people are bleeding all the time. We need surgery. Somebody's chopped off their arm working in some dangerous factory. Battles are being fought all over the world, and gunshot wounds and knife wounds and bayonet, whatever. Okay, people need blood. Okay, and back in the day, people would just bleed out, and there was no hope for them because we didn't really know that we could give each other our blood. Okay, so blood typing became very important because they would see in some cases that was successful and the patient recovered just fine. In other cases, they would have this massive reaction, they would die, and they couldn't figure out why does some people respond well and others didn't. Well, it turns out that the surface antigens on the surface of the red blood cells, is the reason, and because our immune system can detect foreign red blood cells. So let's explain why that is. So we have this ABO system, okay? Those are the names of the antigens that we'll be talking about. And what I want you to think about for this ABO system is that it's kind of like a name tag, okay? This ABO antigen is like a name tag. It tells you who you are, okay? So if I am Josh and I make type A negative blood, every single one of my red blood cells will have a name tag on the surface saying A minus blood, A negative blood, okay? And therefore my immune system will see the name tag and say, yep, that's my human, and I'm not gonna attack it because that's my blood, okay? the immune system recognizes your own blood. But if your own immune system recognizes somebody else, like B positive or O negative, you say, hey, that's foreign. Okay, that's not my human, that's some other human's blood. And so there's going to be a reaction to that. Okay, so, but donating blood, one of the most important things in the clinical setting, because again, accidents happen all the time, and anytime you have surgery, that's an open surgery typically, if there's any blood loss, there's trauma, people are coming in from car accidents or whatever, okay? Blood donation is very important. We can't synthesize blood, okay? It's a cellular thing. Only a donation from another human really helps solve it. So there are 4 basic blood types, plus or minus the RH factor. We'll talk about RH factor in just a moment, okay? So really there are 4 blood types, but really there's only 2 types of antigens, okay? 3 if we include RH factor. We have the A antigen and we have B antigen, okay? And then we can have both A and B together, or we can have neither A or B, okay? So here's a nice convenient table. In the top row is showing you what is the antigen that is expressed on the surface of the red blood cell, okay? If you're a type A blood, you're expressing type A antigen. If you're type B blood, you're expressing type B antigen, okay? Conveniently, red and blue, blue for B, that kind of helps. AB, okay, you are an individual that is actually expressing both types of antigen on the surface. And O, you are expressing no antigen, which is really a misnomer. We'll get to that a little bit later. But there's not an A or a B. Okay. The next row down here is showing you what your antibodies are in your body. Okay? Your antibodies. Antibodies are this Y-shaped protein that your immune system creates in response to, in preparation for, detecting foreign blood. Okay? So if you are a type A, meaning you're expressing A antigen, your immune system will make antibodies against B, because B is viewed as foreign. Okay? So if a type A person received B blood, your immune cells and your antibodies would attack it, okay? And you'd have a bad outcome. B type blood has antibodies that are anti-A, okay, against A or bind to A. If you have AB, that means you don't have any antibodies against A or B because both are claimed as self, okay? So no antibodies against either of those. If you don't have any antibodies expressed on your red blood cells, then your immune system will express antibodies against both A and B, okay? So there's this nice symmetry to this table if you understand what's going on. Okay, so who cares? Why do we need to pay special attention to blood type? Because if you donate the wrong blood, you can have what's called a transfusion reaction. Okay? A transfusion reaction occurs when a type A person receives type B blood, or O, or AB. Okay? Now, there's some unique combinations there that are safe, and we'll talk about that a little bit. But this is what we want to avoid, a transfusion reaction. Okay, because once the antibodies recognize the foreign blood, it will literally rip those cells apart, and then you'll have a big problem. Okay, you can run this type of an analysis. You can run it on a little plate. You add the antibodies, and it will cause a precipitation reaction. Okay, this is what these antigens actually look like. The antigen is the A or the O antigens. Okay. So here's the plasma membrane, and here's this glycoprotein, okay, N-acetylgalactosamine, okay. N-acetylgalactosamine is, there's different names for these sugars. It's basically a modified sugar that's dangling off of the outside of your cell, okay. Here's your cell. Here's a picture of the red blood cell, and it's displaying a very particular name tag, okay. Type A looks like this. Type B looks like this. There's a different combination. I don't care that you know what the sugars are. And then type O, there's still something there in type O, but it doesn't have this particular location occupied. Okay, so it's recognizing this whole molecule, but this one has it missing. Okay, we just refer to this as type O. It's not A and it's not B. And if you express both of these, well then you're type AB. All right, now what about RH? Okay, RH is also called antigen D, but we don't call it ABDO, we just call it RH. And you either have RH or you don't, okay? RH factor. You're either RH positive or you're RH negative. But we just skip the RH, we just say A positive or A negative, or O positive, O negative, right? Now, there's a special relationship that can occur between a man and a woman when they are producing a child, okay? That has to do with the RH factor, okay? It doesn't happen in the ABO because that's taken care of with genetics, but the RH factor is unique, okay? I'm going to walk you through what that scenario is. It doesn't happen all the time, and even if you happen to have the right combination of man and woman, if you know your blood type, you can handle this very easily, okay? So don't get freaked out. It is managed quite well. So let's walk through this diagram. Okay, you have an RH positive man, okay, indicated by these red pluses, and you have an RH minus or negative woman. And they fall in love and they get together and they have a baby. Because each offspring is going to have a combination of the man's and the woman's genes, this baby will have, potentially have, RH factor, okay? So depends on which allele gets donated in the sperm or the egg, right? If his sperm has the positive allele versus the negative, it just depends on his genetics. Okay, but at least the potential of the baby has RH factor and mommy does not. Okay, therefore mommy could potentially have an immune reaction. Okay, let's walk through that. Okay, so first pregnancy. Okay, first pregnancy, we have a baby that is RH positive, and mommy's immune system can become alerted to the foreign blood of baby. Okay? That's the first exposure, so the immune system is naive. It doesn't really know what's going on. It says, hey, this looks foreign, but I don't even know how to handle that yet because I haven't developed antibodies. Okay, so after that first pregnancy, mommy's immune system starts to develop antibodies that are against that RH positive protein or antigen, okay, because she saw it during labor and delivery. Usually there's a little bit of bleeding when the placenta is removed. There's some ripping and tearing of some of the tissue. It's a normal part of the delivery process. The afterbirth is when the placenta gets removed. It's how the baby is attached to the inside of the womb. Anyways, during that procedure, the baby's blood and the mother's blood can mix just a little bit, enough to give an immune response. The baby's out, the first baby is out, so it's protected. It really doesn't have any effects on the first pregnancy, okay? No problem. However, in the meantime, between the first pregnancy and the second pregnancy, mommy's immune system has now generated an immune response, okay? And so mommy is now carrying antibodies. So for the second pregnancy, okay, if we, again, with daddy, that's going to give the RH factor, that second pregnancy has a potential problem, right? Because now mommy has antibodies that could attack the foreign RH factor. And these antibodies can cross the placenta. They can go from mommy's blood into baby's blood, okay? And that's important, okay? Some antibodies can cross the placenta. Okay, and then baby would be a goner because if you have that immune response. Okay, so that can cause problems for mommy and baby. However, if we stop this initial exposure, okay, of the first pregnancy, we can use a treatment called RhoGAM. Okay, RhoGAM is a method of blocking mommy's immune system from exposure to that RH factor. Okay? RhoGAM can block and really shield or cover the eyes of your immune system, and the immune system is blind to RH factor. So it never develops the antibodies, and therefore mommy can have multiple pregnancies with RH positive babies, and there's not going to be an issue because mommy's immune system has been blinded by RhoGAM. So we can stop the adverse effect. All right, so that's Rh factor. We'll talk more about that in class. If you have some questions, we can go over it. Not every single pregnancy with Rh positive, Rh negative, and not every woman and man has to worry about it. If you're both negative or if you're both positive, no problemo. Okay, it's only if the man is positive and the woman is negative. All right, this table is just showing the U.S. average, okay, OAB, AB, and whether you're Rh positive or negative. So the average across the U.S., okay, most people are going to be O or A, okay, 46% versus 40%. Very few people, 10%, are B, and even fewer people are AB, okay, 4%. Okay. However, the other thing I wanted to point out is that the Caucasian population has the highest percentage that is Rh negative. Okay? Everybody else around the world, okay, Hawaiian, Filipino, Chinese, Japanese, Korean, okay, South American, Australian, Aboriginal, all of them are Rh positive. But Caucasians and to a lesser degree African Americans have some Rh negative folks. Okay? So 15% of Caucasians and about 5% of African Americans are Rh negative. Okay? So that's just kind of an interesting factoid. Okay? It's about how prevalent is this across human populations. Korean Americans tend to be pretty even, 30, 30, 30, 10. Okay? You don't have to memorize that. I just thought it was kind of an interesting factoid. So most common blood type, O, okay? O positive. O positive is the most common type. The most rare type would be AB negative. AB negative. I believe I am A positive. Although I haven't gotten it checked recently. All right, so that's really red blood cells. That's the first of the formed elements, okay? You have three types of formed elements: red blood cells, white blood cells, and platelets. We've just covered red blood cells, okay, and all the things that red blood cells do. We talked about hemoglobin, talked about anemia, talked about the life cycle of red blood cells. Now, let's talk about white blood cells, okay? White blood cells, there are 5 types of white blood cells, okay? They're listed here. We're gonna get really friendly and be able to recognize the differences here, okay? It's easy, actually. I'll give you some tips about how to recognize them. Okay, leuco means white, okay? Erythro means red, leuco means white. No hemoglobin. These cells do not carry oxygen, okay? That's not their job. Okay, they have nuclei and other organelles, and their 3 main things that white blood cells do are to defend, to remove, and to attack, okay? So I like to think about these 3 categories of immune response, okay? Defend against pathogens. Think about foreign cells. If you have bacteria, okay, like E. coli. E. coli is a cell, it's a foreign cell. So we're gonna defend against a foreign invader, okay, like a virus or a bacterium. We can remove toxins, okay, so there's not just cells, but actually foreign molecules. So it's proteins or other molecules can get into our body, and that can create a problem. We have cells that can protect us against that. And then lastly, we can attack abnormal cells, okay? What I like to call bad self, meaning I have cells inside of me that could go rogue, meaning I have cancer cells or other cells that are not behaving like they should, or other cellular parts that originate from me, okay? And our white blood cells can be trained to identify cancer, okay? Now, of course, not all cancers can be detected, okay? That would be wonderful if we could detect all cancers by our own immune cell, okay? It's not possible. So foreign cells, foreign molecules, and bad self, okay? Virally infected cells are also would be considered bad self, okay? Most white blood cells are in the tissues, okay? So tissues is bigger, more white blood cells live in the tissues than they are circulating. Okay, so they float around in the blood a little bit, but once they float around, they'll migrate from the blood into a tissue, the muscle, or to the GI tract, or into the wherever. Okay. So there's 4 characteristics here of white blood cells. Okay, circulating white blood cells. They can migrate out of the bloodstream. Okay, they start in the bloodstream and they can actually move in and out of the bloodstream. This process of moving in and out of the bloodstream is called diapedesis. Diapedesis. We'll talk more about diapedesis. The ability of a white blood cell to float around in the bloodstream and say, hey, I would like to get out of the bloodstream, I'm going to hop into the tissue. White blood cells can do that. Okay? They have amoeboid movement. They kind of blob around and they can make their way through tissue. Okay? They can push their way through the extracellular space and in and through tissue. Number 3, they're attracted to chemical stimuli like chemical hormones, which we call positive chemotaxis. So think about here, this cell is being attracted to some stimulus here. Okay, it's like, guys, if you smell a lady's perfume, you're like, wow, that smells great. And you kind of are attracted towards, where's that smell coming from? I love the smell, I need to find the girl that has that smell. That's kind of a loose, analogy for white blood cells can pick up on the scent, as it were, or the chemical stimulus from some attractant, okay, that's recruiting this cell to a particular location, like the site of an injury. You have all kinds of chemokines that could be recruiting, saying, hey, come over here, and they use chemicals to do that. It's called positive chemotaxis. Positive means you're moving towards the stimulus. Chemotaxis, taxis means movement. You're moving towards the chemical. Okay? Negative chemotaxis would mean you're moving away from that. It's like it's a danger signal, like get away from here and run far away. 4th one here is some of these white blood cells can perform phagocytosis. Okay? Phagocytosis is cellular eating, right? This is a picture of a white blood cell eating up some bacteria. Okay? Think about Pac-Man eating up something. Phagocytosis turns out to be one of the most effective mechanisms for your immune system to mitigate some foreign invader, okay? Some pathogen. Okay? So phagocytosis, very, very important. It can gobble up foreign cells, foreign molecules, and bad cells. Moving in and out of the bloodstream, okay? They have this amoeboid movement, okay? Positive chemotaxis, phagocytosis. So here's a picture of a common injury. Okay, here we have somebody getting a splinter or a scrape or a cut or an abrasion. Any type of compromise to the integumentary system would cause the outer layers of the epidermis and dermis to be compromised. Okay, bringing along with it, because bacteria are everywhere. Every single surface, there are millions and billions of bacteria. And so if you have an opening in your skin, pretty good chance that you're going to get some bacteria in there. Once they're in there, these little black rods, you could say, then there's resident macrophages or other immune cells that can detect, hey, you don't belong here, and they recognize the bacteria, and then they start to release chemokines, and that will signal to the local bloodstream, a capillary bed, say, hey, there's a foreign invader here, sound the alert, recruit more help. We need some soldiers to show up on site and beat off, okay, to battle against this foreign invader. So these mast cells or resident macrophages can communicate, and then these cells can migrate through the cell wall here. It's called diapedesis. They're floating around here, they migrate through the cell wall, okay, through the vessel wall, I should say. The cell wall, the vessel wall, and they show up and they begin to gobble up the foreign invader, the bacteria, okay, using phagocytosis. Once those cells, which are primarily, the first cells to show up are usually neutrophils. Okay? Neutrophils show up first, and they are committed to their job. They show up, they gobble up as many bacteria as they can, and then they kind of die. They die in place. And that's the formation of a lovely substance that we know as pus. Okay? Pus is that ooey gooey white stuff that we typically see in an infected lesion, okay, or wound. Okay? When you see pus, you should say, hey, there's a whole bunch of white blood cells that have just died defending me. Okay? There's a whole bunch of white blood cell soldiers that have showed up to the battle and they've done as best they can to gobble up as many bad guys and they've died in the process. So if you have a lot of pus, that means that you have a lot of white blood cells that have been working to mitigate the infection. Okay. And then so once we've stopped the bacterial invasion, then we can work on healing the skin. All right. So here's just some terminology that you need to know. Neutrophils are the most common response to any type of bacterial infection. Okay? So they're going to be tumbling along the blood vessel, okay? And they will actually detect these chemical signals. Okay? Chemotaxis, there's these blue molecules here that are functioning kind of like a hormone. This would be a paracrine mechanism. Remember, endocrine is molecules being communicated through the bloodstream. Paracrine is really through the interstitial space or through the tissue. Okay, paracrine function. Autocrine is one cell to itself. Paracrine is like short distances. So these chemokines are recruiting the neutrophil. The first part of this is that the neutrophil finds its way against the edge. Okay, that's called margination. Okay, margination means it gets stuck to the edge. It stops flowing along with the rest of the red blood cells. Okay, and then after margination, we have diapedesis. Diapedesis allows this cell to actually insert its hands and kind of pull open an opening in the endothelial cells, okay, the walls of the blood vessel. And it kind of squeezes its way through, and now it can move towards the site of injury, okay, wherever this chemokine is being signaled from. Think of chemokines as being like a 911 call, like, hey, you guys, I need some help over here. And these are responding to that call. All right, so those are some characteristics of all white blood cells. We talked a little bit about phagocytosis there, okay, and amoeboid action. So how to distinguish white blood cells? When we look at all of these, how are you going to know what is an eosinophil versus a lymphocyte? Okay, we're going to look at the size of the cell. We're going to look at the color. We're going to look at the shape. Okay, we're going to look at the granules. Do we have granules, yes or no? Okay. Now there's a joke here. The color and the shape is the name of one of my favorite albums from high school, the Foo Fighters, one of the best-selling albums, rock albums of the alternative rock scene in the late 1990s. Okay, the color and the shape is the name of the album. That's why there's a joke. Anyways, okay, so we're going to go through how to identify these different cells. Okay, the first thing that we're going to recognize is really the color. Okay, now there's two types of stains that we stain tissues with. There's kind of a bluish color, which here looks like purple. That's called hematoxylin. And then we have a reddish pink stain called eosin. Okay, H and E, hematoxylin and eosin. All you have to know is that hematoxylin is blue, it turns out purple, and eosin is pink. Okay, eosin is pink. So when we stain cells, the blue hematoxylin is attracted to the nucleus because it's staining negative charge. Okay, and DNA has that negative phosphate backbone, so hematoxylin staining the DNA. Okay, eosin is attracted to positive charge, so the cytoplasm is typically pink. So when we look, so basophilic means blue, basophilic is a word that means it is attracted to a negative charge. Okay, basophilic, blue, hematoxylin. Eosin, okay, eosinophilic means pink. Okay, that means it has a positive charge. Now, we're going to go through each of these. Okay, neutrophils, basophils, eosinophils, monocytes, and lymphocytes. Where are we going to start here? Remember, these are all a mixture inside of the blood. 99.99% of all of your blood cells are red blood cells. But that small little percentage are these white blood cells. And they're all over the place. Okay. When we look at a picture of a stained slide, we can see immediately regions of blue or purple and red. Well, we know this, okay, really, really pink and really only pink. Those are a whole bunch of red blood cells. Okay? All of these are little red blood cells. Here we see only blue or purple, okay? These happen to be lymphocytes. Okay? Lymphocytes have a prominent blue stain, large nucleus with respect to the cytoplasmic volume. So these are all lymphocytes. Okay? And then we have some scattered, here's probably a part of the capillary here. So very quickly you can tell the difference between different cell types just based on color. All right. So if we talked about over here the pathway for differentiating red blood cells. Now let's talk about these over here, okay? And the way we're going to memorize them is to understand something about their lineage, okay, where they come from. So there's three here, which we call the phils, okay? The neutrophil, the basophil, and the eosinophil, okay? If you remember what we just talked about, these stains, basophil means blue, so B for B, basophil means blue. Eosinophil means pink. Now, forget for a moment this looks orange. I don't get why the artist colored it kind of an orangish color. It should be pink. Okay? Neutrophils are a neutral stain. It's not especially blue, it's not especially pink, it's just kind of neutral, kind of pale gray. Neutrophils. What are they filled with? They're filled with granules. Eosinophils, basophils, and neutrophils all have granules. A granule is a speckled appearance within the cytoplasm, and these have a lot of granules. So therefore we call these the granulocytes. Granulocytes. They all have phil at the end, okay? Basophil, eosinophil, neutrophil. What are they filled with? Kind of play on words. They're filled with granules. Those granules are the speckly appearances, have a speckly appearance. These are the agranulocytes, meaning they lack granules. And therefore you're not going to see a speckled cytoplasm, you're going to see a kind of a evenly stained cytoplasm. So the really super easy way to memorize your white blood cells is to remember your three phils. Okay, you have a really, really pink one, the really blue one, and then there's a neutral one. Okay, so pink, blue, and neutral. Okay, eosinophil, basophil, neutrophil. And those are kind of medium size. Okay, then you have big guy and little guy. Okay, big guy is called a monocyte. Okay, it's a very large cell and very large nucleus. And they have the smallest of the five, which is the lymphocyte. Okay, so three phils, big guy, monocyte, and little guy, lymphocyte. Little guy, lymphocyte, L for L. So done, you have it memorized, okay? You can visually do that by the color and the presence of granules and the size of the cells, okay? But we're gonna get some more practice on that. All of these cells are controlled by their own set of hormones, okay? But they're called CSFs, okay? CSF stands for colony stimulating factor, okay? Colony stimulating factor, CSF. Do not confuse this with cerebrospinal fluid. Remember CSF in the brain? If that combination of letters sounds familiar to you, like CSF, cerebrospinal fluid, that's like the fluid in the brain, right? Yes, there's a different CSF, okay? Unfortunately. Colony stimulating factor. Okay? It just means it's a hormone that is triggering a very particular step in the differentiation of white blood cells. All right, and there are more generic hormones and there's more specific ones. Okay? So if you have a CSF, that's colony stimulating factor. If you have an M in front of it, that's the monocyte colony stimulating factor. If you have a G in front of it, that's a granulocyte. We talked about those. Those are the phils. GCSF. You have a GM-CSF. That's going to be the combination of the granulocytes or monocytes. So it's a more generic one. Or the multi. Okay, it's the most generic. It can stimulate all of the downstream cell types. Okay? Any of these could be stimulated by the multi-CSF. Now, just a reminder, all of this is happening in the bone marrow. Okay? White blood cell production and red blood cell production is happening in the bone marrow, okay? In the spongy bone of your vertebra, of your long bones, okay, of your clavicles, primarily in your sternum and your vertebra into adulthood. Vertebra and kind of your coxal bones, your hip bones, contain the highest amounts of red marrow. So So if we were to zoom in on the red marrow, this is what we would see, okay? There's a bunch of kind of undifferentiated stem cell tissue that's in various stages of differentiation. You have a blood vessel or capillary that's running right through the red marrow, okay? So here we could have a pathway for making neutrophils, okay? This is the precursor stem cell and it's making neutrophils, okay? You could go off in another direction and be making eosinophils or basophils or whatever, okay? Here's a macrophage. Here is something called a megakaryocyte, okay? Because in just a little bit, we're gonna talk about platelets, okay? Platelets are little bits of cells that are derived from this big mothership, okay? It's a big mother cell, it's huge, and it's sticking its fingers into the bloodstream and it's breaking off these little tiny bits of cytoplasm which become platelets, okay? So the megakaryocyte, mega means big, karyocyte is the rest of it. Again, a very similar diagram. It's the same thing, I'm just showing you another version of it because it looks pretty. Another diagram. This one helps me remember to think, to remind you about monocytes, okay? Here's your red blood cells, here's our platelets. Megakaryocyte makes platelets. Here's our three phils, okay, our granulocytes, eosinophil, basophil, and neutrophil. You have a big guy and little guy. Now there's two little guys here because you have B cells and T cells. Okay? But this is where I want you to kind of pay attention at this point. Monocyte is the biggest of the 5 white blood cells, okay, in its size. It has a very large nucleus, and its nucleus happens to be kind of a kidney bean shape, okay? It has a slight C shape to it. When monocytes navigate from the blood into tissue, it changes name. The convenient thing is it keeps the M as the first letter. Monocytes become macrophages, okay? A monocyte in the blood becomes a macrophage in the tissue, okay? Any tissue, okay? If it migrates out of the bloodstream, okay, going from blood into the tissue, it becomes a macrophage. Macrophage means big eater, okay? Macro means big, phage means mouth or to eat, phagocytosis. Lymphocytes here, we'll talk more about those in a subsequent chapter. You have T lymphocytes and you have B lymphocytes. B lymphocytes can be differentiated into plasma cells. These are your antibody factories. Mast cells come up from a very similar lineage here, the myeloid stem cells. Mast cells, we'll talk about those in the immune system as well. Mast cells have a very similar appearance as basophils because they're both very, very blue. Okay, again, very similar diagram. If you've seen one of these diagrams, you've seen them all. They have a very similar flow to them. This one I like because it's showing you actual images, like photographs of these cells. One thing I want to point out here with neutrophils is neutrophils have the highest segmentation of its nucleus. Notice that many of these nuclei have more of a round or at most kind of a kidney bean shape, but neutrophils often have 4 or 5 discrete segments. That is, they're pinched off in between and they form these discrete segments. 4 to 5 segments, that's a mature neutrophil. Early on in the differentiation, it looks kind of like a monocyte, okay? It has just kind of a kidney bean shape, but it begins to become segmented, okay? This is called a band cell, okay? A band cell will be further segmented into a mature neutrophil, okay? And then mature neutrophil, it almost looks like you have discrete, different nuclei, but they're all actually connected. All right, so a little bit more on some of these details about neutrophils. Notice that we have listed all of the 5 here. Now I'm going to go through one by one and start telling you some specific things that you need to know about each of these. Okay, we're going to start on neutrophils. So what I just described is that neutrophils have a very distinct and unique nuclear morphology. Okay, that's fancy words for it has a really special looking nucleus. Morphology is this the shape or the appearance of something. 4 to 5 segments, okay, very distinct. So if you see a cell that has a bunch of segments, automatically neutrophil, Okay? These might have one or two, okay? Or they're just kind of generically kidney bean shaped, but the basophils and eosinophils are not highly segmented. Okay, 50–60% of circulating white blood cells are of this type. Often, neutrophils are referred to as polymorphonuclear sites. Polymorphonuclear leukocytes, or sometimes referred to as PMNs. Okay? PMNs, if you look at a pathologist's write-up, you'll see PMN, okay? Polymorphonuclear sites, cells. All right, so we talked about how a mature neutrophil has a very highly segmented appearance. When they're immature, they're going to appear as like a band, meaning they're becoming kind of elongated and kind of curling on itself, but it's not quite segmented, meaning pinched off. So this is called a band cell. And it means that this neutrophil is young. It's on its way to becoming a mature neutrophil. So when you see lots of young neutrophils, you might be on the front edge of a bacterial infection, okay? Because your body's been alerted to it and says, hey, we need a bunch of neutrophils, so your body starts to make new neutrophils. And some of those early neutrophils might not be quite ready yet, and they may have a look of a banded appearance. We call them band cells, just as an indication of young neutrophils that have been called into action. Okay, so band cells versus segs, meaning segmented. All right. So neutrophil action, very active, first to attack bacteria. So if you have a bacterial infection, you know who you're going to call. You're going to call the neutrophils. Okay? They engulf and digest pathogens usually using phagocytosis, and they have a process called degranulation. Okay? Degranulation just means those granules get ejected and opened up, and the granules contain enzymes and other chemicals that can attack the foreign invader. Okay? You also have defensins, which are special proteins that they're peptides from the lysosomes. And if in a really bad case, okay, if a very special case, you have something called oxidative burst. Oxidative burst is when the cell releases a whole bunch of oxidative products that does a special kind of, redox karate chop, and that kind of kills everything in its wake. It's kind of like what's the thing in the sci-fi movies when they hit the button and they send up this electrical burst, okay, a magnetic pulse. It it renders all the electrical stuff like it can't work. That's kind of like what oxidative burst is. Alrighty. Also, the formation of pus. Okay, pus, the white gooey stuff that forms an infection, is dead neutrophils. All right. That's where I'm going to break for now because I'm out of time. We're going to start off with the eosinophils next lecture, and the next lecture we'll finish off blood.